Hello, I'm Ben Samelson-Jones from the Children's Hospital of Philadelphia and the University of Pennsylvania School of Medicine. And I recently had the privilege of presenting at the American Society of Hematology annual meeting. On behalf of my co-authors, follow-up of more than 5 years in a cohort of patients with hemophilia B treated with Fidanacogene Elaparvovec, Adeno-Associated virus gene therapy. In this abstract, we report the longest follow-up to date of hemophilia B gene therapy recipients expressing the high specific activity factor 9 variant, R338L. Fidanacogene Elaparvovec, which was previously known as Spark 9001, is a hepatotropic bioengineered adeno-associated virus, or AAV, vector that delivers the high activity factor 9 variant, R338L, under the control of a liver-specific promoter. We have previously published the short-term safety and efficacy of this vector in the first 10 participants in 2017. In this abstract, we report the long-term experience out to 5.5 years of these initial recipients, as well as 5 additional subjects. All recipients received 5 times 10 to the 11th vector genomes per kilogram in a phase 1-2 dosing study, and then were eligible for enrollment after 1 year in the long-term follow-up protocol. 14 out of the 15 recipients participated in this long-term follow-up, of which 13 have ongoing enrollment. Fidanacogene Elaparvovec was generally well tolerated over the follow-up period. No participant developed a factor 9 inhibitor or experienced a thrombotic event. No participant required treatment with corticosteroids in the long-term follow-up study. Serious adverse events were reported in 3 participants, including a major traumatic injury, but none of these serious adverse events were considered to be related to the gene therapy vector. Annual surveillance by liver ultrasounds did not demonstrate any hepatic masses, and there was no significant elevations in alpha-fetoprotein. 13 of the 14 participants, 93%, with available data had a factor 9 activity levels in the mild or normal range for the last assessment prior to the data cutoff, with a mean activity levels of 23% normal at year 1 and between 20 and 25% normal for the next 4 years. Importantly, these factor 9 levels were measured in the central laboratory with an actin FSL-based one-stage clotting assay. The mean annualized bleeding rate was less than 1, with 80% or more of the participants having 0 bleeds every year out to year 6 of follow-up. 7 participants underwent 9 surgical procedures without bleeding complications. 2 were emergent procedures, an appendectomy and a lumbar discectomy, and were performed without additional factor 9 treatments. Prior to the other 7 procedures, factor 9 replacement was administered, as would be typical for mild hemophilia. In conclusion, the long-term expression of factor 9 R338L after a single administration of Fidanacogene Elaparvovec is sustained for at least 5 years with an annualized bleeding rate of less than 1. This treatment was well tolerated without safety concerns, including thrombotic complications despite major traumatic injuries and emergent procedures. There is a larger phase 3 study that is currently ongoing.